Hello, and welcome to GradCast, the official radio show and podcast of the Society of Graduate Students here at Western University. I'm your host, Ariel Frame.
And I am your co-host, Laura Munoz.
And we're here today with another member of the lab that I work in. So I'm a friend of mine and a coworker of mine that I'm really glad to have on, Asad Lone, PhD in biology, working on his PhD in biology, senior PhD. Welcome, Asad.
Hi guys, good to be back. Yeah,
that's right. You are an experienced in the GradCast experience, and this is your second time on the show. So we'd like to know what you're working on generally, and maybe you can just dive basically right in with what you've been doing since last time you were on the show.
Yeah, so just to give everyone a brief overview, I do work on a disease called Alzheimer's disease, and Alzheimer's disease is a neurodegenerative disease. It's the most common form of age-related dementia. There's like 50 million patients around the world, and 10 million new patients get diagnosed every year. So it's a really important epidemic in the sense that it's really it's widespread around the world. So what we work on in our lab, in the coming lab, we focus on metabolism in Alzheimer's disease. And someone might ask, why do you look at metabolism? Because the dominant hypothesis, or the main focus for the last 25 years in this field has been focused on a protein called amyloid beta. The hypothesis called the amyloid cascade hypothesis, which posits that it is the buildup of toxic protein in the brain as we age. The protein is called amyloid beta, and it's a sticky protein. It's when you look at it under the microscope, it looks like this starchy protein called amylin, and it makes these plaques, extracellular plaques within our brains, which can cause a lot of synaptic loss and neuronal death. So, which then causes a lot of memory deficits, personality deficits, and then eventually leading to a very painful decline in health. Sorry. So, why look at metabolism when everyone focuses on amyloid beta? I think there is quite a lot of evidence to support that the main causal factor in Alzheimer's disease might not be amyloid beta. It actually might be the metabolism of the brain. So one of the main things that not a lot of people know, is that as we age, brain metabolism declines as well. So when you take a brain of a healthy 30-year-old compared to someone who's 70 years old and completely fine, who doesn't have Alzheimer's disease, there's a decline in brain metabolism. But when you take someone with Alzheimer's disease, compare it with someone who's 30 years old, there's a significant difference that you can see in the glucose metabolism of the brain of someone who's 30 years old compared to someone who's maybe like 75, 80 years old and has Alzheimer's disease. And so we kind of want to try and understand what proteins are involved in what proteins are involved in controlling the metabolism in the cells in our brain. And so that's what my kind of project focuses on. Is I look at one specific protein called P66 and we kind of treat it as a switch. We turn that protein on, we turn that protein off, and we look at how that affects the metabolism in the cells of our brain. So, so basically, what we talked about in our last podcast was that the protein that I work on, the paper that I published since, we showed that this protein acts as a molecular switch that can change the metabolism of a cell from a mitochondrial-based metabolism to glycolysis. And I'll explain that in a more digestible manner. So the mitochondria, think of the mitochondria as the engine of the cell, or the more commonly known as the factory of a cell that is there to produce energy for everything that the cell does. And you know, just like an engine, when you have a fuel, in this case that is glucose, you produce energy, but at the same time you also have emissions. Like, you know, a car running on gas, it produces a lot of power, but it also has a lot of emissions. And that is kind of like what the mitochondria is. So the cells of our brain, they're very energy demanding. The brain is 2% of our body weight, and it uses up almost like 25% of all energy produced in our body. So it's very energy demanding. And so the cells of the brain, they primarily use mitochondria um, to produce energy. And then there's a lot of pollution that is produced as well. However, there's another way that the cell can make energy, and that process is called glycolysis. And it's actually just fermentation of sugar. It's basically you take glucose and there's a pathway of enzymes that converts the glucose to lactate. And then if there's any hydrogen ions around, they react with this molecule and they produce lactic acid. And most people might know lactic acid, you know, when you're running on a treadmill, and after a while you might get off and you have pain in your muscle, in your legs, in your calves, and that's actually lactic acid buildup. It's, you know, when you run on a treadmill, you're gasping for air, and your cells are using the muscle cells in your legs are using mitochondria to produce energy, but the mitochondria needs oxygen. And when you run really fast on a treadmill, you need to be inhaling a lot more oxygen than, you know, for example, me, I'm just sitting down on a chair. And at some point, the amount of oxygen going in is not enough for all the cells in our muscles to be able to produce energy required to run on the treadmill. And so how does the cells in our muscles kind of make up that energy deficit? They switch to glycolysis, which is basically it takes glucose in, it doesn't require oxygen. So it's basically millions of years ago through our evolution, bacteria, they could take in glucose and they didn't need oxygen because maybe oxygen wasn't there in our atmosphere, and they could take in sugar and they could ferment it and they could produce energy. Our cells have still maintained that method of making energy, but a byproduct is lactate, and that lactate reacts with hydrogen, and then you get lactic acid. So I know I'm gone a little bit off topic, but just to explain these two different ways of making energy. So think of a hybrid car that can use gas and electricity. So gas would be your mitochondria or your mitochondrial metabolism taking in, producing energy, but you have a lot more emissions. But glycolysis would be when you run out of gas, you would switch the car to your car battery. It's electric now. So you're still producing energy, but you're not producing a lot of emissions. So it's a cleaner way of producing energy. And the protein that I'm working on acts as a molecular switch to switch going from mitochondrial metabolism to glycolysis. And so people might ask, why is this relevant in Alzheimer's disease? Well, so my supervisor, Dr. Robert Cumming, his work, his postdoctoral work in the Salk Institute, they actually showed that if you have cells being cultured in a dish and you add that toxic protein amyloid beta that builds up in the brain that most people think that is causing Alzheimer's disease, when you take a dish of neurons, cells from the brain, and you put that toxic protein on it, most will die, some will survive. And if you take those cells that are surviving and then you keep growing them, you know, for generation after generation, with that protein, that toxic protein acting as a environmental stressor, those cells will eventually kind of be completely they'll be completely what's the word I'm looking for, damage proof. You know, the amyloid beta won't be able to kill them. And so what Robert did, my supervisor, is he looked at what's different in those cells, the ones that are not, that amyloid beta cannot kill, than the ones that amyloid beta can kill. And they found that the ones that are resistant to amyloid beta, they actually use glycolytic metabolism. So they used a clean form of energy. They don't use mitochondrial metabolism. So that was quite a significant discovery. But at that time, we didn't know, how does that shift happen? How does a cell switch from using mitochondrial metabolism and then go to this more cleaner form of energy called glycolysis? And so part of my initial project, where my first publication was we identified one of the proteins involved in this switch. And I showed that when this protein is switched on, when it's phosphorylated, this protein can change the amount of enzymes in the cell that are used in different types of metabolism. So if this protein is on, the cell is using mitochondrial metabolism. And so the levels of the enzymes that are used in glycolysis, they go down. But when you turn this protein off and you switch this metabolism of a cell towards its more cleaner version, the level of those enzymes actually go up. So the enzyme expression increases. And we also showed that when the protein is switched off and it moves to this glycolytic metabolism, there's less pollution. So there's less production of what we call reactive oxygen species, or ROS for short. And so there's less ROS, which means that the cell is under less stress. And then finally, we wanted to ask the question that if all these beneficial effects are occurring when you turn this protein off, are these cells actually more resistant to the toxic protein amyloid beta? So I took a bunch of cells and I transfected them with an siRNA, but we don't need to talk about all these protocols. But basically, I switched that protein off and I added amyloid beta into this dish of cells. And what I found was that these cells were actually more resistant to amyloid beta. Far fewer of those cells died than the ones that had this protein switched on. And so what I showed was that when you have this protein switched on in neurons, the cell is using mitochondrial metabolism. There's a lot more pollution. There's a lot more ROS in these cells. Even though they're producing more energy, that might not be beneficial in the long term because there's a lot more stress that these cells are under. And then when you add amyloid beta on top of that, the cell, there's a threshold as to how much stress a cell can undertake. And when that threshold is crossed, the cell dies. And so when a cell is using mitochondrial metabolism, the threshold, you're almost at that threshold. But then when you add an environmental stressor such as amyloid beta, the cell's like, okay, I'm done. I can't handle this much stress. I'm going to tap out.
So
but when
I wonder, sorry.
No, go ahead. Yeah.
I wonder usually how does the cell clean the pollution that is produced by using mitochondrial, like using the mitochondrial way to produce energy? How is usually that pollution is usually cleaned?
Yeah, that's a good question. So the cell cells are amazing because they almost have an answer to every problem that they create themselves. So there are these enzymes, these proteins that a cell makes, and they're called antioxidant enzymes. So how a lot of people say that, eat a lot of blueberries or eat berries or eat whatever new food, superfood that's in fashion because it's an antioxidant. That's what they're talking about, that you're giving your cell the prerequisites, the building blocks to make these proteins, these antioxidant proteins. And their function is kind of like recycling. What they do is they take these damaging species, these reactive oxygen species, and there are different ways that they can neutralize them. And so what happens is that when a cell is going under some kind of stress, it will activate an antioxidant pathway, which is a series of events that happen leading to these proteins called transcription factors binding to our DNA and promoting the production of these antioxidant enzymes. And then these antioxidant enzymes can go and kind of like make sure that these damaging molecules, these reactive oxygen species, are taken care of so that they don't damage other proteins and they don't damage other DNA. So the cell has an answer to all these problems. So yeah.
So
Yeah, go ahead, Ariel.
So Assad, you've characterized the way they can, these cells can adapt to different stressors over time, and the one that you found and characterized most deeply was this P66 Schick, and how it's bad news on
Yeah.
its own. You don't want to
It's very
have it.
bad news. Yeah, yeah. In
So
some study, in earlier studies, they show that if you take a mouse and you switch this protein off, basically you silence the expression of this protein. These mice live longer.
So
And
that's a
one of
connection from the cells, because you work with the cells like you keep saying a dish. So you're literally growing cells individually in a dish, but these are cells from a multicellular organism like us, or like a mouse, or like a rat, where they live all together. And you're showing that even in the whole organism, if you
yeah.
have if you take it away, then these animals fare better. So I'm just trying to see if like we can look at back to a connection. If Alzheimer's disease is a disease of humans and not other animals. So do you see now that you've done a lot of the characterization on the molecular level, and that's all the work that you've now published on P66 Schick? What's the next step to get to the human disease?
Yeah, that's a good question. So, there are other groups that have worked on this protein as well in the context of Alzheimer's disease. So, one of the conferences that you and I actually went to, the Alzheimer's disease conference in 2017, I believe it was in Toronto. I met one of these. Luckily, I just met one of the scientists who was working on this same protein in Alzheimer's disease, and he's based out of Switzerland. I can't remember his name right now, but what he did was basically he took these animals, these a disease model that we use to study Alzheimer's disease. These are basically mice that have specific mutations, specific genes that have been put in their genome, which allow them to get the same type of deposition of this protein in their brain that like humans do. And so what he did was he took these Alzheimer's mice and he bred them with mice that had the P66 protein turned off. So what he ended up with was a generation of mice that had that were going to have a lot of amyloid beta in the brain, like like an aged human would, but it didn't have that protein, the P66 protein. And when he did a battery of memory tests on these mice, he actually found that these mice were actually a lot better in remembering or having better memory than mice that had this protein switched on. And so there's already data that exists that shows that when you turn this protein off, at least in animals, they actually have better memory compared to animals that have this protein switch on. So for a preclinical data perspective, there is data that already exists. And so I think there needs to be more work done in animal uh, models. And I think that once we have a safety or a better depth understanding of what this protein is doing when you just switch it off, I think that we can start investigating this protein in humans as well,
Yeah,
because
that's
safety comes
actually
first.
that's actually my question, because I was wondering, because as you said before, the cells are so wise and they know what they're doing most of the time. So there must be a reason why this protein is turned on or off. So
Yeah.
like if we just turn it off, or because it's better for Alzheimer, maybe we're screwing a lot of other things in the cell.
Yeah, that's a great point. And that question keeps me up at night, right? Because, you know, if like I don't think that anything in there's no kind of molecular pathway that's redundant that you just take away or you put it out or you switch it off and it will have no effect on the body. Like 100%, if you switch this protein off, it's not all good news. There are going to be some consequences to that. If anyone ever comes to you and says that, oh, I look at this protein and I have made this discovery and it's all good news, no bad news, never trust that. you know so one of the things that I worry about is that for this P66 protein, because it when you switch it off, some of the downstream effects that happen, even though they might look good, one of the things I worry about is that they might be actually pro-cancerous. So what is cancer? Cancer cells actually are cells that are growing really rapid. They grow fast. And interestingly, cancer cells use the same kind of metabolism that makes a cell resistant to amyloid beta. So the question is, if you take a neuron and you switch its metabolism to one that is protecting it against amyloid beta, are you potentially making it cancerous? Right? So these studies need to be done in mice where we have an animal, and you switch off P66 Schick and you let them live. Is there do these animals have a higher incidence of cancer or tumor growth or something like that? So these studies need to be done. That's why in the beginning I said the safety. We need to look at whether this is safe. But in humans, and I'm talking like really down the line, if this protein ever becomes a drug target, to switch this protein off, I don't think that it would be lifelong. It would either be something that would be very targeted in a short period of time. So it would be someone who just got an Alzheimer's disease diagnosis. Okay, maybe we can give this specific whatever type of drug it would be to maybe for a short time switch this protein off and see how it would affect that person. Would it improve memory? Would it help with cognition? Like, these are the questions that we need to down the line try and answer.
And how is
So
that possible? How can you target just one protein
Oh, so
for a
that's
period of
that's the
time?
hot stuff these days. It's
Oh,
called
okay.
precision medicine. It's
Oh,
being used in cancer a lot. So if you have if you have an oncogenic protein, which is basically a protein that can promote the production of a tumor, there already are a lot of drugs and clinical trials that specifically focus one protein in like a pathway, and they've done a lot of these studies in humans, and I think there are quite a few FDA approved drugs as well that specifically target one protein. So that technology is there. We just need to get there for Alzheimer's disease. Yeah.
so let's say we wanted to do the best we could while somebody somewhere works on some precision medicine. I think a lot of people think when they think of metabolism, they think of like eating, like it's food. That's what I take food in, and that food becomes like smaller food for my cells,
Yeah.
so that
Yeah.
my
Yeah.
cells
Yeah.
can then eat. So I'm
Yeah.
eating, putting in my mouth is by proxy putting food into the mouths of all my cells. So
Exactly. Yeah.
is diet related to this phenomena of like glycolysis versus the mitochondria versus like which our cells are switching to? Does diet have any impact here, and what are your thoughts on
Yeah.
diet and Alzheimer's?
So that's a topic that I'm really interested in, and I personally think that diet plays a significant role in all chronic diseases. And if you actually read the literature on Alzheimer's disease, then insulin resistance or diabetes is a huge comorbidity that is linked with Alzheimer's disease, to the point where Alzheimer's disease is actually called type 3 diabetes. So the brain actually becomes diabetic where it cannot use glucose for fuel. So how people there's this misunderstanding that people say that glucose or sugar is needed by the brain. That's actually completely false. In terms of need, glucose comes in number three. It comes in third in what kind of food the brain prefers. The number one is actually ketones, and number two is lactate, and comes in at number three. So the ketogenic diet is really famous these days, gets a lot of heat, but in uh, from a therapeutic point of view, it's a very strong intervention, and quite a few randomized clinical trials have shown that the ketogenic diet can actually improve memory and cognition in patients who are suffering from Alzheimer's disease. And basically, it works through a couple of different ways. But and to answer your question in a brief manner, if the brain cannot use one fuel, just give it a different one, because all the cells in our bodies, our brain, our muscle cells, they can use different kinds of fuel, just like a hybrid car can use gas and electricity. Our cells can use ketones, lactate, glucose, these different molecules to make energy. And if a brain cannot use glucose, if it's, quote unquote diabetic, if it's insulin resistant, then just give it another fuel. And that's basically how the ketogenic diet works in the context of Alzheimer's disease. It provides these energy molecules called ketones that the brain cells can take up and produce ATP, or the energy currency in the cell, and thereby the brain cells are not starving. They're functioning normally then.
And which kind of food can you eat to get these ketones?
Yeah, so a ketogenic diet is a high fat, low carbohydrate diet. And so what you have to do is you have to focus on reducing your intake of carbohydrates, which are mainly you processed carbohydrates. So you want to eat less rice, bread, sugar, the bad stuff. And then you want to increase, you want to eat more meat, and you want to eat more butter, cheese, vegetables, the good stuff. So it's basically almost like a healthier diet when you compare it to the standard American diet. So it's a high fat, low carbohydrate diet. Yeah.
Very cool. And makes me wonder, like, I mean, why don't people just do that now? You tried this out yourself?
Yeah, I mean, I've even taken part in a clinical trial on the ketogenic diet. I love taking, measuring different parameters on my body, and, sleep, or blood glucose, blood ketones, and so when this opportunity came up that I could take part in a clinical trial, and I'd be getting a blood test like every 2 weeks, and I could look at my LDL and my HbA1c and, cholesterol. And so I was like, yes, sign me up. Collect as much data as I can on myself. Yeah.
That's so cool. It's nice to see that you're passionate about this topic, and you really adopt it into your day-to-day life as well. Where does the day-to-day life of Asad Lone go after their PhD? You're almost done here. You're writing your thesis at the moment. So what are your plans?
Yeah. So initially, I was like, when I started out my PhD, I was very passionate about that. I want to stay in academia. I'll do a postdoc, and I'll somehow weasel my way into like becoming a professor, or a scientist in a research lab. 5 years later, I'm not that hot on that idea anymore. I think that I need a bit of a break from academia, and so what I'm thinking is I'm exploring my options as either in consultancy, or in some form of more broad role in like pharmaceutical, maybe even in public health. With what's going on these days, I've been kind of really interested in like public health data and the responses to such crises. And so I think that these are some of the roles that I'm kind of interested in these days. And that's one good thing about doing a PhD, is that because you're picking up so many different skills. You're not only a scientist, you have to be a writer, and you have to travel, and you have to attend conferences, and you have to manage time, you have to manage expenses, you have to design experiments. That almost makes you a problem solver in different areas. And so I think that's one thing that I want to kind of pursue, use these real world skills that you get as a PhD student and apply them in a different environment other than academia. And maybe I'll retire in academia, who knows.
Cool. So just our last question here. If people wanted to like find you online, where's the one place they could go?
Yeah, so I share my research on ResearchGate. So if you just search for Asad Lone, you'll find me there. I'm usually like the top link that comes up in Google. And you can also check out our lab if anyone's interested in studying metabolism or Alzheimer's disease. You're more than welcome to check out thecominglab.com. We share all our new publications. We share the kind of work we do. You can contact students. You can contact our supervisor. So it's a great website to get resources on what we do there.
Excellent. Thanks, Asad. It's been great having you on the show. This has been Gradcast, the official radio show and podcast of the Society of Graduate Students at Western University. I've been your host, Ariel Frame. My co-host was Laura Munoz, and we've been speaking with Asad Lone. This episode is also produced by Laura Munoz, co-produced, co-host, and producer. If you'd like to get involved in the show, get in contact with us at gradcastradio@gmail.com. You can also follow us on Instagram, Facebook, Twitter at gradcastradio. You can listen to us on the radio, Radio Western 94.9 FM. All our episodes are on our website at gradcast.ca, and you can just find us as wherever podcasts are available, basically, as well, including Spotify. People like Spotify nowadays. We're there. Alternatively, the video version of a lot of our episodes are on YouTube. That's also Gradcast Radio. Thanks for listening. Have a great night.